A cool, milky liquid slides into a vein in your arm. In about 40 seconds, it reaches your brain. It will not put you to sleep, and it will not switch you off like a light. It quiets your neurons, one by one, until your brain can no longer hold the constant conversation that adds up to you. And just like that, you are gone. General anesthesia is a drug-induced, reversible state, a brain guided out of consciousness and back on purpose, under close control. That control starts at the level of a single molecule, so follow one drop of the drug down to one nerve cell. On the surface of that neuron sits a tiny gate called a GABA-A receptor. Normally, it opens only when the brain's main calming signal arrives, letting negatively charged chloride ions flow inside and making the cell harder to fire. Propofol grabs that gate and strengthens the calming current, holding it open longer so even more negative charge pours in. The neuron's voltage sags below the level it needs to send a signal. Do that across billions of neurons at once, and the brain goes quiet. A few anesthetics take a different route. Ketamine, for one, plugs an excitatory gate called NMDA instead, but the result points toward the same end. Excitatory traffic is disrupted, and the network can no longer sustain ordinary awareness. But quiet neurons are only half the story. One leading explanation for what dissolves your awareness is what those neurons stop doing together. Deep in the center of your brain sits the thalamus, a relay hub that trades signals non-stop with the cortex, the wrinkled outer sheet where perception and thought take shape. Being awake seems to depend on that back-and-forth traffic, millions of messages a second, binding sights, sounds, and thoughts into one connected scene. As the drug takes hold, the messages thin out. The loop slows. The cortex and thalamus stop answering each other, and the single unified picture that is your consciousness breaks into disconnected islands. There is no one off switch in there. Awareness fades because the conversation that builds it can no longer hold together. You can actually watch clues to this happen from outside the skull. An EEG reads the summed electrical rhythm of the cortex and draws it as a single wavy line. Awake, that line is fast, busy, and irregular. As anesthesia deepens, it changes character. A strong, slow rhythm rises up front, and big, lazy waves begin to carry the trace. Push deeper still, and the brain can reach burst-suppression. Seconds of near silence broken by short flickers of activity. The shape of that line offers a live estimate of anesthetic depth, one of several signals the anesthesiologist may watch. Here is something most people never realize. There is rarely one single anesthesia drug. Full general anesthesia is a cocktail because it has several separate jobs. One drug switches off awareness. Another blocks the brain from forming memories, so the procedure leaves no trace. A third stops pain signals before they ever reach the brain. And often a fourth, a paralytic, freezes the muscles so the body holds perfectly still. Each of these acts on a different target, and none of them alone is unconsciousness. That is why the anesthesiologist works like a pilot, not staring at one dial, but constantly reading depth, breathing, heart rate, and drug levels, and adjusting the mix minute by minute. Separating those jobs is what makes modern surgery possible. It also creates one rare, unsettling failure. Because paralysis and unconsciousness come from different drugs, it is possible, in rare cases, for the muscles to be frozen while awareness has not fully gone. The body cannot move or signal, even though some thread of the mind is still there. Doctors call it intraoperative awareness. How often it happens depends on how you count. Older studies that interviewed every patient afterward found it in roughly 1 to 2 people per thousand. Counts based on patients reporting it on their own come out far lower. It is more likely when a paralytic removes the usual clue that anesthesia has gone too light: movement. This is rare, and modern monitoring has made it rarer. Watching the brain's own rhythms can help, though no monitor is a perfect mind reader. It is one instrument among many. So how do you come back? For a long time, the assumption was simple. The drug wears off, its level in the brain drops, and you wake. But waking is not a clean rewind. Coming back is an active reboot. A 2025 review highlights a striking clue from animal work. As the brain climbs back toward consciousness, cells in one part of the thalamus rapidly strip away a protein called KCC2, which had been helping keep them quiet. Remove it, and those neurons regain their spark, and the thalamus-cortex conversation begins to flicker back on. This is early science, one mechanism, not the whole story, but it fits something every patient knows. You do not snap awake clean and clear. Different parts of the brain come back on different clocks, which is why waking from anesthesia feels foggy, heavy, and slow before you are fully yourself again. We have been performing this vanishing act for a long time. In 1846, in a surgical theater in Boston, a dentist showed a room full of doctors that a breath of ether could leave a patient unresponsive through surgery. It looked like magic then. Nearly 2 centuries later, we do it millions of times a year, with remarkable safety and control.
And here is the honest part. We can reliably switch a mind off and switch it back on. We can measure the drugs, watch the waves, and bring almost everyone safely home. But we still cannot fully explain the deepest thing in the room. Why all that electrical traffic between the thalamus and the cortex feels like anything at all, like being you. Anesthesia turns that feeling off and on with a needle and stands quietly at the edge of the biggest question in science. Modern anesthesia is remarkably safe and closely watched. If you ever face it yourself, the people to talk to are the professionals who do this every day.